Hi, my name is Alyssa Casey, and today I will be talking about membrane receptors and signal transduction. Alright, so what I have drawn here is going to be our phospholipid bilayer. That is going to be the blue portion. The upper part of our video is going to be the extracellular portion, and the bottom part of our whiteboard is going to be the intracellular portion. Now, what I'm drawing here is going to be our receptor. What the receptor does is basically it is going to be the communicator of the cell. It's going to take anything that any neurotransmitters or hormones that might come its way and attach to the receptor in order to communicate with the cell what the body is going to need or any processes of the body. So right there I've drawn what I call a ligand. That is going to be our neurotransmitters or our hormones. So as you're going to see, when this ligand moves over and attaches to the receptor, it's going to communicate with the receptor what the cell needs to do and hopefully produce a cellular response. Now, what I'm drawing here is going to be our G protein. Now, this G protein is going to have, it's called a heterotrimeric G protein, which means it's essentially going to have three parts. It's going to have the alpha subunit, the beta subunit, and the gamma subunit. These are all very important parts in communicating to the cell what is going to happen next. Mostly we're going to be working with the alpha subunit. The alpha subunit is basically going to be telling the cell what it's going to be doing. So we have these two things. There's GDP and GTP. These two are very important because it's going to essentially say what the alpha subunit is going to do. Now, if we have GDP attached to our alpha subunit, essentially it's going to be inactive and nothing's really going to happen. So signal transduction does not continue. So instead, if we have GTP connected to the alpha subunit, then we're going to continue in process. Now, what GTP is going to tell the G protein to do is to communicate with something called adenyl cyclase. Adenyl cyclase is something that is going to communicate, or the G protein is going to communicate. The G protein is what we call a first messenger, and so this first messenger is going to communicate with adenyl cyclase in order to create cyclic AMP, which we'll talk about in a minute. Real quick, I just want to mention that sometimes instead of adenyl cyclase, we're going to be working with phosphodiesterase, which is the absolute opposite of phosphodiesterase, or of adenyl cyclase. So instead of building cyclic AMP, phosphodiesterase is going to tear down cyclic AMP. So what I'm drawing here is the ATP. So what adenyl cyclase does is it takes ATP and then it's going to create cyclic AMP. So through this process, it's going to end up just creating lots of cyclic AMP. It's basically enhancing the response, so there's going to be an increase in cyclic AMP in the cell. Something that cyclic AMP does is it's going to communicate with a protein kinase. So as you're going to see, I'm going to draw over here. And basically what a protein kinase does is it causes an action in the cell. So in order for a protein kinase, specifically today we're going to be talking about protein kinase A since it is very specific to cyclic AMP. Protein kinase A is going to cause a reaction in the body. Or not in the body, in the cell. So it's going to cause a reaction in the cell. So you're going to see that that cyclic AMP is going to bind to the protein kinase. Realistically, there's going to be a lot of cyclic AMP, and there's going to be a lot of ATP turned into cyclic AMP, and there's going to be a lot more protein kinases, but I don't really have enough room for that. Okay, so we're going to see that our cyclic AMP binds to protein kinase A. what protein kinase A is going to do is it's going to activate possibly our phosphorylation process. So as you can see, this little blob is going to represent our phosphorylation. So we have phosphorylation. Woohoo! What phosphorylation does is it helps transfer a phosphate group off until an action is produced. Specifically, I'm going to talk about glycogen. This is assuming that this is a process going on in the liver. So we're going to take glycogen and it's going to go through the process of phosphorylation, and the cellular response, which is basically the whole process of signal transduction, and the whole goal of signal transduction is a cellular response. We're going to take it and it's going to create glucose. But that's basically what signal transduction is, and the whole idea of what membrane receptors